All right, so everyone make it back from lunch, hopefully somewhat dry. Maybe not. It's okay. Did everyone make it back from yesterday? That's a first. That's great. I'm just kidding. No, everyone shows up the second day. At least gotta make it a week where you fall. I'm just kidding. So any questions from the first section from yesterday we covered? So you guys are all experts at it now. Just understand what the body does. Maybe we'll get into more details. Don't you worry about that. So we'll finish up section 1 and then we'll head into section 2. So have those, and everyone's in the Canvas course, correct? Everyone can see it. Everyone saw the link to the YouTube, hopefully, so you can get to that if you need to rewatch anything. So that's available to you as well. I'll typically do that. Usually try within the same day to get those videos up, but give me 24 hours or so before you start emailing and complaining. Some people are like 2 hours later, where's the video at? It's like, hold up, it takes a little time. Anywho. Going back to, we were talking about acid-base status and how, we have pretty tight ranges on where that needs to be within the body. Again, we said a normal serum pH 7.35, 7.45. Not necessarily that you memorize that number specifically, just know we do keep it within pretty tight ranges there. So the question is, how do we keep it in that tight range? And we need buffers, right? So what do buffers do? Like if you go take your car and they kind of wax it. Yeah, go ahead.
They pretty much counter whatever acidity is in your blood.
Yeah, that's a good way to explain it. So basically, it tries to prevent kind of wide shifts in hydrogen ion concentration, hydroxyl ion concentration, in order to keep things within that stable pH, right? And so typically, you're going to find that most of the buffering capacity comes from the blood, right? Especially with a lot of this kind of balance between CO2 and bicarbonate is going to be, especially within the red blood cells here. So you can kind of see that when we have energy production that's happening within the cells, usually a byproduct of that is going to be what? CO2, right? So again, by utilizing oxygen, we're going to convert that over to CO2. And CO2, is that good for us, bad for us? Within range, it is okay, but we need to get rid of it, right? And the primary place we get rid of CO2 is where? The lungs, right? And so the blood's going to be the main thing that's going to be transporting that carbon dioxide up into the lungs, and then we can breathe it out, right? So that process for transporting that has to occur via some enzymes, some enzymatic processes here that occur. And so basically you have this this reaction here that converts carbon dioxide. You'll mix it with this H2O. There's an enzyme called carbonic anhydrase that we'll talk about later on. There's a lot of drugs that actually interact with that. But that'll turn into carbonic acid, and that carbonic acid can actually dissociate into bicarbonate and then a hydrogen ion, essentially, right? And so what you end up finding is that when you have too much hydrogen ions around, we said when you have an abundance of hydrogen ions, what does that do to the pH? Drops it down, right? So it's going to make it more acidic. So when you have a large amount of hydrogen ions on this side of the equation, where's it going to force that equation to go? Yeah, back in the opposite direction, right? So it's going to go more over to producing a lot of carbon dioxide. And so you have to keep this balance, and bicarbonate is really useful for that. So bicarbonate is going to interact with that additional hydrogen ions, and then it can eventually turn to carbon dioxide and be transported out and breathe it out. And so we'll talk more about that, especially in regards to the kidneys, because that's going to be one of the other big places where we're going to be managing bicarb and managing acid-base status. But the lungs are the other big place where we're going to be expelling a lot of that CO2. And again, normally you'd find that normal serum pH is, the normal range. If you go too high, you're too basic, you call that alkalosis, and you're too low. Acidosis, absolutely. And again, you don't want to get too far outside of those ranges because that's when death occurs. Typically, you want to keep your patient outside of these ranges most often, right? Right? Hopefully you guys got that down at least. And so, you can find different sources for an acidosis or an alkalosis. Typically, it can either be of a respiratory or a metabolic cause. So if you ever hear someone talk about a metabolic acidosis or a respiratory acidosis, you can find that it's helping you to diagnose where the actual problem is. So for instance, if I'm holding on to too much CO2 because I'm not breathing well enough, that would be called like a respiratory acidosis, right? And so you'll get into that in other classes later on and get a better idea of kind of where those are coming from, what's the actual pathophysiology causing that in the first place. A big thing that I deal with very frequently, actually I was on call for the Poison Center last night. I had a case that was kind of like this, where if you have someone who's taking too much opioids, anyone familiar with opioids? What's an example of an opioid? Oxycontin's a really good one. Any other ones? Morphine, fentanyl, heroin, all the good stuff are opioids. Use it for pain control in a lot of cases. But if you take too much of that, you end up stopping you stop breathing essentially, right? So the patient stops breathing, they end up developing a hypercapnia. They have too much CO2 in the blood floating around. We call that a respiratory acidosis that ends up developing because of that. So again, just one reason why you may develop that. Lots of other reasons, but you'll get into that later on. So just an example. All right, some other molecules we'll deal with are going to be our carbohydrates. What are carbohydrates good for? Energy production, absolutely right. So again, it's going to be a large organic molecule. You're going to find that we'll talk about different types of carbohydrates, whether they're more simple or more complex. Lots of varieties out there. What are some examples of carbohydrates you may have just taken in during lunchtime? Glucose, just starches, like things like in breads and whatnot. What about if you're drinking milk? Lactose, right? So there's all kinds of different sugars that are out there. They're very important for us. Mainly, you're going to find that they typically end with the suffix OSE. It's like glucose, lactose, sucrose, any of those would denote that, yes, it is a carbohydrate. Main source of energy, especially very quick sources of energy. We'll find that you can also get this energy from other sources like proteins and fats, but this is one of the fastest ways we can do that. So again, very important for us. So yeah, just a single sugar, what do you call that? A monosaccharide versus, say, if you had two, disaccharide if you had many, polysaccharide, absolutely. So you guys already know that. And again, typically these have to be broken down into more simple forms in order to be, one, digested, typically, and also be utilized for things like energy production and producing things like ATP. So for instance, we have enzymes that are going to be able to break these down. So for instance, when you, say, take a bite of food, what's one of the first things that kind of interacts with the food in your mouth? Saliva. And anyone know what's in saliva? Amylase, absolutely. So amylase is really good because amylase is actually going to be responsible for starting that breakdown of the carbohydrates into simpler forms that we can absorb a little bit easier. And so it's very important. We need those enzymes. If not, then we have malabsorption that occurs, and you don't get those good nutrients. There's just some examples of monosaccharides you might run into. So here's glucose, fructose, galactose. All these have different roles in the body. Typically, glucose is going to be one of the main ones that we're going to be concerned with as far as dealing with things like hypoglycemia, hyperglycemia. Glucose is that big thing we're measuring with that. Okay, some disaccharides, things like sucrose, again, they typically need to be broken down into their kind of constituent forms in order to aid with absorption or aid with utilization in different enzymatic processes. We mentioned polysaccharides. Glycogen can actually be considered a form of a polysaccharide. We mentioned that glycogen is used for what?
Break down glucose.
Yeah, so we use it as storage. Say we have excess blood glucose, we can utilize that and take it to the liver and convert it into glycogen, and then it can be utilized for storage, and it can be broken down later on in that process. Remember what we called that? Yeah, glycogenolysis. You're breaking it down, and then you use that for gluconeogenesis, right? So those two processes go hand in hand there. So that's a good one. Cellulose is where you find the main constituent there in plants. Lots of different varieties we're going to deal with. But glycogen is a very important thing, especially when you're dealing with patients who may have low blood sugars. Typically have low glycogen stores as well. A lot of that has to go with their carbohydrate intake and different factors like that. So here's an example of what glycogen looks like. It's just a big long chain of sugar molecules that we can break off and utilize to produce new glucose as needed. And again, that process mainly happening in the liver. You'll find it happen in some other places as well, but the liver is one of the primary sources of gluconeogenesis. And that's really important because if you're inhibiting that, or if you have any kind of, say, deficiency in glycogen, you can end up developing things like hypoglycemia. And that could be, a reason why your patient's, say, having a syncopal episode or developing fatigue or things like that. Next, we have our lipids, and so lipids are typically, we kind of mentioned before, we talk about polar molecules and nonpolar molecules. We talk about things being hydrophilic, hydrophobic. The other kind of side of that coin there is going to be when we talk about things that are going to be lipophilic or lipophobic, right? And so that's going to be an important term. We're going to talk about that a lot in pharm. We're going to talk a lot about it here as well. But this is where we're going to be seeing our lipids here. Typically, things that would be lipophilic tend to be they tend to solubilize pretty well in lipid solutions. So here's kind of the baseline form of them essentially. And so notice here, this is a fatty acid. Why do we call it a fatty acid? You know where the acid comes from? Any organic chemistry nerds out there? Yeah, this carboxyl group right here, that's the acid portion of it, right? So these fatty acids can be utilized because, again, we're going to see with energy production, you need carbon essentially to produce things like ATP. With sugars, you get that carbon very easily from that, but this is another place we can do that. That's why long-term storage of carbohydrates tends to eventually turn into fat storage, essentially, right? So this is where you get a lot of that carbon sources there. And again, if you think about it, like if you look at the calorie per gram of food, and of course, dietitian, don't give it away, please. If you're looking at, say, a gram of carbohydrates, anyone know how many calories there are in a gram of carbohydrates?
4.
Yeah, we'll learn that later, but basically it's 4. If you look at a gram of fat, anyone know how many calories are in that?
9.
Actually 9, yeah. So there's actually a lot more energy stored up there in a single gram of fat or lipids. But again, it's not going to be as quickly used. And we'll get into that when we talk about the nutrition section a little bit later. But fatty acids are important. Again, we're going to see that this is where a lot of things like steroids are going to be made, things like testosterone, estrogen, all that end up coming from a lot of these fatty acids. So again, let's see. We talk about saturated versus unsaturated fatty acids. What does that mean when I say unsaturated? Yeah, there's double bonds that are available. Why are double bonds useful?
Structure.
Hmm?
More stable structure.
Not necessarily stable structure, but they can be utilized for different chemical reactions. Usually when you have like a saturated fatty acid, you're going to find they're not very or as useful, and that's why we talk about foods low or high in saturated fat. But basically you're going to find that all that's referring to is whether or not there's double bonds that are available acids here. But anyway, just another example where we or another thing we'll actually measure. So you're dealing with patients, say, has high say cholesterol. What do you call that? It's kind of the medical term for high cholesterol. Hyperlipidemia, right? So one of the things you actually measure for those patients is called triglycerides. Have you heard of triglycerides before? Basically, this is another storage form for our lipids where you're going to take these 3 fatty acids and you actually mix them with the molecule glycerol. This will come up again when we talk about energy production from fat utilization later on in the section here. But basically, when you have a triglycerol or triglyceride, this is where you're going to see glycerol mixed with these 3 fatty acids. And these can be liberated individually and can be utilized for things like energy production or hormone production, things like that. So you'll definitely see those come up later on. Again, just an example of a saturated versus unsaturated. Here's an example of a saturated fatty acid. Notice there's all single bonds here between all these carbons versus here the unsaturated fatty acid. There's going to be some double bonds there. Okay. All right. Ketones are also really important. So these are going to end up being formed when you have hydrolysis of these triglycerides. So again, you start to break them off, and you're going to have these free fatty acids that are floating around in the blood. So anyone ever heard of like ketones or ketosis? Anyone know when that occurs? It can happen with high blood sugar, namely in patients who are diabetic, right? What do you call that? Yeah, diabetic ketoacidosis, so DKA. That usually is going to happen in your type 1 diabetics. Anyone know when else you might produce ketones?
Starvation.
Yeah, starvation, absolutely. So if you're going like on a keto diet that's low in carbohydrates or you have a starvation state, that is also where you're going to be generating a lot of ketones. That makes sense because if I don't have carbohydrates to use for energy, where else am I going to go for energy? You're going to go to the fat, right? It's there for storage anyway. So you start to produce these free fatty acids. And so they form into these ketone bodies that are there. You can also have things like acetone that ends up getting formed. So again, you think acetone, where do you normally see that? Say like in the household. And nail polish remover. Your body can actually produce that as well. And that's actually one of those things, anyone never smelled acetone kind of has what kind of smell to it? Yeah, a little bit of a fruity smell. Other ketones will have that fruity smell as well, and that's why if you have a patient who has DKA, or fulminant DKA, they may actually have a fruity sort of odor to their breath. A lot of that is due to the ketone bodies that are being formed there. But just know that ketones get formed in these kind of sort of states. We'll talk about those again later on, but we just want to get you at least some somewhat of a familiarity with them right now. Again, we mentioned uncontrolled diabetes is a big place we see a lot of ketone bodies being formed. That kind of makes sense because from an insulin standpoint, we'll talk about this later on when we talk about insulin in more detail. The insulin is really necessary in order to get glucose into your cells. And with a diabetic patient, if they have a lack of insulin, all that blood sugar, all that sugar is in the blood and it's floating around there, but it can't really get into the cells. And so because of that, your cells have to convert over to using fatty acids, and that's why they have this ketone production that happens with that. Again, don't get too dragged down on that. Just know that we're going to talk about that later on. But the problem with ketones and these free fatty acids in the blood, what do you think that does to your pH? It's going to lower, right? Because again, they're acids themselves. They can liberate those hydrogen ions, and so that's why those patients develop a diabetic ketoacidosis because of those fatty acids that are there, right? It's one of the reasons. So you can have a patient with DKA that comes in, I said normal pH 7.35, 7.45. These patients come in 6.9, right? In some cases, really severe case, and it's pretty significant. So we have to do things to try to alkalize the blood, try to get the pH back up to a normal range. But a lot of it is due to those ketones production, the ketones that are being produced there. This is another really important thing here. These phospholipids are kind of considered a form of lipids here. Where do you normally see a lot of phospholipids? Anyone know? Cell walls, absolutely. So we have this lipid bilayer. It's right here. And why are lipid bilayers so important?
Keeping things out of the cell.
It keeps stuff out of the cell that should be out of it. It keeps stuff in the cell that should be in it.
It forms a barrier between polar and non-polar, so isn't that the idea?
Absolutely, yeah, perfect. So basically what you're finding with these lipid bilayers, there's a polar head right here. You can see this, and it's usually made of like a phosphate group with some stuff added onto it. We'll see some stuff that can be included with that. But you also have these fatty acid tails. And notice that the fatty acids are going to be kind of joining up together. Because again, one of the big things I'll try to hammer home a lot, especially in the farm course, is that like dissolves like. So fats like to be in fats. Water likes to be with water. Things like that. So what you normally find is that in these kind of aqueous solutions, right, because again, your blood and extracellular fluid tends to be a kind of a more aqueous sort of water-based solution, that polar head is going to want to orient itself to that, right? And if you imagine this is within the cell, those heads are going to polarize there, or at least face that direction as well. Here you're going to have this kind of this fatty core that is going to be kind of preventing a lot of things from getting through. Yes, sir?
If it's hydrophobic, is it unaffected lipophilic by default, or are the two related or not?
Yeah, absolutely. Those two are absolutely related. So I have something that's very hydrophilic. It tends to be very lipophobic for the most part, right? And so again, you can kind of look at those as going hand in hand. And so that's important because again, if I have something that floats around in say water, so say for instance you have something like a molecule of glucose. Glucose itself, solubilizes into water, right? So anyone have like a soda here with sugar in it? Like it goes into solution, right? Pretty easy. Well, that glucose is not going to be able to just cross into the cell that well because it's one of those things that's going to be too, it's not going to cross that lipid bilayer because it's going to be repelled by these kind of lipophilic sort of forces here. Now, certain fatty substances are going to be able to cross that very well, and we'll talk about membrane transport a little bit later on and things that can cross via passive diffusion. Things are going to require transporters. We'll get into that a little bit later. But just know lipid bilayers are very important because it keeps stuff outside of the cell that should be outside and keeps stuff inside of the cell that's there, right? If you lose that lipid bilayer, what happens to the cell? It loses its integrity. Imagine if you lost your skin, what would happen? You'd probably die, right? So the same thing happens to the cell. And that's important when we're looking at things like, say, for instance, with antibiotics, right? So when you're looking at bacteria, we want to kill off bacteria. Typically, if you have an infection, we can give things like antibiotics that actually pierce a hole through that cell wall, open it up, and then the cell dies. All right, so it's important we maintain that integrity. Otherwise, the cells are going to develop, or they will die. It may develop things like apoptosis. You guys heard of that term before? What's apoptosis? Yeah, programmed cell death. And we'll talk about that a little bit later. But again, that's one of those things where you typically want that integrity intact there. Okay, looking at a phospholipid, you can see here the fatty acid chains are going to be coming off of that. This would be the lipophilic portion of it, right? So again, it likes to be with lipids, does not like to be with water. So again, this would be very non-polar versus the head here. This phosphate group tends to be much more polar. Right, so again, it's going to go into solution in water very well. It's going to be more of the hydrophilic portion, right? So again, another way to say this is lipophobic, this is hydrophobic. Does that make sense? Okay. The other big thing you can see with a lot of phospholipids is that we actually use that. We will liberate phospholipids from the cell membrane in order to generate things like hormones or steroids. And so you'll notice that these steroids tend to be pretty fatty sort of molecules. And anyone know any examples of steroids? Testosterone's a big one, right? The main male sex hormone. What else? Roughly half the population will produce. Actually, all the population will produce estrogen, but progesteron's also be another one. There's a lot of different steroids that we're going to talk about, especially when you talk about things like cortisol. What does cortisol do for us? Ever heard of that?
Crazy sugar.
Yeah, it's like a stress hormone, right? So again, it's secreted by the adrenal glands. All of these are going to be structurally very similar to one another. They usually contain this sort of cholesterol kind of based core here. And you can see they'll generate lots of different things based on whatever enzymes are interacting with them. So for instance, if you're in the adrenal glands, you'll generate things like cortisol. However, you can also produce things like progesterone. Androstenedione is actually something that's like a precursor to testosterone. Lots of different things can be formed from that. But just know they're being ultimately generated from these lipid bilayers. Those fatty acids there that can be generated into this cholesterol can eventually turn into these steroids. So they all share sort of a common sort of beginning in that regard. Even things like, cholecalciferol, vitamin D. What's vitamin D used for? Yeah, helps with calcium absorption, helps to maintain good bone health, things like that. So that's also kind of again kind of falls in this category of steroid here. But lots of steroids we're going to use in medicine, especially when you're dealing like inflammatory conditions. We'll talk about those pretty much ad nauseum later on in the pharm course. So just know we're going to talk about these again. Okay, and then prostaglandins is going to also be a type of fatty acid that gets formed here. So this is prostaglandins are very important, and we have several ways of modifying this, especially with medications. But they are going to help with various things depending on the organ system you're talking about. So for instance, prostaglandins can help with regulating things like blood vessel diameter. So this can help with like blood flow to certain areas, like namely the kidneys, because again, kidneys you want to keep good blood flow going to. Prostaglandins can help with things like ovulation, uterine contractions. One of the biggest places you're going to see, and if you ever have like a headache or you have like a strained muscle or something like that, what do you normally take for it at home? So if you have a headache, so there's Tylenol, which is one type of drug, but what's the other thing you might normally take? NSAIDs, right? NSAIDs, like what's an example of an NSAID? Ibuprofen. Probably everyone has that in their home, or at least has taken it at some point in their life. That's actually going to be one of the big things you're going to use as an anti-inflammatory because it actually helps to inhibit prostaglandin synthesis. So there's going to be lots of ways we can interact with this. But just know prostaglandins have several functions throughout the body. We'll talk about them as we get into the individual sections later on. So just kind of get a familiarity with the terms that we're kind of using here. Okay. And again, arachidonic acid is one of these precursors that you actually get from that lipid bilayer. You can kind of notice the lipid bilayer sort of view here. Notice it's going to interact with several different enzymes. Don't get hung up on this right now. We'll talk about these more in detail when we get to where they become important, right? Some of these are important in the lungs. Some of these are going to be important for other places as well. So just know that we're going to come back to this again and again later on. Especially in pharm, this will come up quite frequently in pharmacology. Okay. So the last thing we talked about, so we mentioned carbohydrates, we mentioned lipids. The last one is going to be proteins, right? So our last macronutrient, right? If you think of macronutrients, you think carbohydrates, you think lipids, you're going to be thinking proteins here. So proteins, the other big thing, usually composed of an amino acid chain, right? And basically there's several different amino acids that are available within the body. Some of these, a lot of them we can make on our own. Some of these you're going to find we actually cannot make on our own. We actually have to get through our diet. I'm not going to have you memorize every single amino acid that's there. I'm not that mean because I remember when I was in pharm school and I was in biochem, we had to memorize every single amino acid and be able to draw it on our test. And that was no fun. So I don't make you guys memorize that. Just note though that some of them are going to be things we have to get through our diet. A lot of them we can make on our own. And how do we determine what a protein chain is going to be? How do we determine like what kind of amino acids go to make a protein? It's going to be dictated by our DNA, right? So again, we're going to talk about protein production a little bit later on, but protein production is super important, especially from the DNA standpoint. We're talking about how those interact with one another. So, you know what you call a mean protein or a protein in a bad mood? Amino acid, right? Okay, anyway, so basically they all share a very similar structure here, right? They have the same core. Again, you can kind of see this. Anytime you see R, what does R usually mean in a chemical structure? Well, C means carbon. Yeah, usually some kind of functional group. So R will be something else, right? So R is kind of a general, you know, there's something else that's there. But they use this to kind of illustrate here. They all share the kind of the same core here. So you know, there's an amino group, there's a carboxyl group, and then there's usually an R group. And whatever that R group is, usually will dictate what amino acid it is. So for instance, valine here, you can see it has this structure versus tyrosine here. Again, that is going to show you which amino acid you're actually dealing with, and again how they get introduced into the protein. The sequencing of those will dictate what kind of protein you make, right? Whether it be a protein you find in, say, a hair cell versus a protein you're going to find within, say, a kidney cell. All are going to be dictated by your DNA when we talk about transcription and translation a little bit later on. Okay. And again, you can kind of see how the primary structure of the protein, how they all have this kind of interlinking that occurs here. And again, a lot of this happens during the transcription phase when it gets kind of translated over from DNA. Again, we definitely have enzymes that are going to be able to break down proteins, so don't forget about that. So again, proteases and things like that are going to be very important, especially when talking about the digestive tract. So we'll come back to that later. But proteins are good because they can actually be conjugated with other molecules as well. So you can have a protein by itself that can be useful for various and sundry things. However, you can also conjugate it with things like a lipid. You can conjugate it with a carbohydrate, and that can be very useful, especially when you see this on like cell surfaces. We'll find there's lots of uses. You can use this for that. But for instance, if you ever see something like a protein plus a carbohydrate, you basically call that a glycoprotein, right? You can find this can be useful in certain things like some hormones. You can definitely find this in cell membranes. Glycoproteins can be useful for things like cell signaling. So if a cell comes in contact with another cell, it can say, hey, I'm a human cell, I'm a host cell, versus something like a foreign cell, like a bacteria. So glycoproteins can be used for that. Lipoproteins would just be a protein plus a lipid. Okay, and again, it can also be found as things like carrier molecules. You'll definitely find these in cell membranes as well. And again, what do you notice about the cell membrane that we already saw earlier? Yeah, that lipid bilayer, right? So again, you're going to find that these cells are not just going to be kind of uniform phospholipids all the way around. They're going to be kind of impregnated with these different proteins that are here, right? Some of these can be things like protein channel transporters. These are going to be basically proteins that are going to open up and allow things to flow through. That can be dictated by which protein that is. And again, I mentioned a lot of things are going to be cell signaling. Some of them are going to be just meant for communication with other cells, things like that. So we'll look at those details a little bit later on. Just some other examples of conjugated proteins. You might find things like hemoglobin. You basically will take hemoglobin, bind to this heme pigment, right? And then you end up utilizing it for things like oxygen delivery within the blood, right? So again, hemoglobin is very important. If you don't have hemoglobin, you can't really transport oxygen that well. How about myoglobin? Where do you normally find myoglobin, do you think? Yeah, I normally find that in muscle. That's where a lot of oxygen storage happens within the muscle itself. That'll be important later on when we talk about energy production. Insulin, this actually doesn't have any kind of non-protein component to it, but again, it's going to be very important for regulation of things like blood glucose. We'll talk about that later. Even things like, your blood group proteins. We talk about blood types. You guys familiar with blood types? What are some examples of blood types? A, B, are you positive? AB positive, right? Okay, we'll get to those later. We'll talk about those again. But anywho, that is very important because those carbohydrates are going to dictate what type of blood type you actually have based on antigens you have present there. So, and then again, lipoproteins are very important when you talk about things like when you think of cholesterol. What 3 letters do you think of? What's your bad cholesterol? Yeah, so LDL is the bad cholesterol. HDL, I think it was like the good cholesterol. It's a little more complicated than that, but essentially LDL you consider to be the bad one. That stands for low density lipoprotein, right? So that cholesterol in your blood, a lot of it is made up of these lipoproteins, right? And that's just basically a protein plus a lipid. That's how you get that essentially. But very important for transporting those lipids, getting around to the places where it needs to go essentially. Okay. Again, lots of functions for proteins. Again, structural, so these can be useful in helping to maintain good structural stability on certain types of cells. Enzymes are always going to be proteins, right? So we'll talk about enzymes a little bit later. Things like antibodies. What are antibodies used for? They're going to help with immune function, right? They're going to be able to bind to things that are considered to be foreign. They're going to bind to things that are going to be targeted by the immune system to get rid of it, right? Anyone know what happens when our antibodies start targeting our cells? Yeah, autoimmune diseases, right? So again, we talked to Dr. Ovalle, rheumatologic conditions. A lot of those are autoimmune conditions that are being where our body's kind of inappropriately attacking our cells, and we have lots of drugs we can deal with that. Other things are going to be like receptors. When we talk about receptors causing certain cell signaling processes to occur, and then carriers are also going to be another big thing, helping to move products from outside of the cell to inside or inside back out, essentially. Okay, last section we'll talk about here is going to be basically our nucleotide composition, talking about DNA, RNA a little bit here. So again, one of the 2 big categories you're going to see with our nucleotides are going to be pyrimidines and purines. Okay, and so when you see the pyrimidines, again, when I'm trying to categorize these in my head, pyrimidines I think are actually the smaller structure, even though they're the larger name. Again, I make odd associations, whatever helps you to memorize, to remember this, happens to be at. But the main pyrimidines you're going to see are going to be cytosine, uracil, and then thymine, right? What's the difference between uracil and thymine? Yeah, so we're going to find uracil in RNA only. We're going to find thymine in DNA only. So we'll talk about that in more detail in a second here. But you also have the purines, typically guanine and adenosine. Adenine. those two are going to be your purines. And again, these are going to have special bonding together, and they're going to help to make up that DNA. They're going to see these hydrogen bonds that form between these. And normally these are going to be bound to a type of sugar. So for instance, with DNA, what's the sugar that these are going to be bound to? Deoxyribose. And then we're going to see that for RNA, it's going to be just ribose, right? So ribonucleic acid versus deoxyribonucleic acid. All that goes back to the sugar that you're actually binding to. And you notice here with ribose, you have this hydroxyl group here. Notice that when you deoxy it, you take out the oxygen molecule, now you have deoxyribose, right? That's where you get your DNA from. This is going to be really important from a standpoint where when we're dealing with cancer, right? What is cancer? If you had to sum it up. Yeah, kind of unfettered cellular replication, right? So for instance, if you have, say, something like leukemia, you're going to have your white cells are going to be replicating kind of just without stop, right? And so we're going to find that when we talk about things like mitosis and meiosis and all of that, DNA is going to be a crucial component of that, and a lot of our chemotherapy drugs are actually targeting the DNA here. So it's important to kind of understand how these things are kind of linking up together to see how things like, medications or things like radiation or things like surgery are going to be able to help to kind of interact with these disease states. So if you're interested in cancer, we'll talk a lot about that later on, and how this can, the DNA is a main target for a lot of those therapies. Typically with DNA or this deoxyribonucleic acid, you're going to find that A to T are going to be the main things that bind there, and you're going to have C and G. Right, notice these kind of hydrogen bonds are going to be situated here, and they're complementary bonds, right? So again, a pyrimidine binds to a purine. Again, are these, not covalent bonds, because again, covalent bonds we mentioned are pretty permanent, right? We want hydrogen bonds here because we want to be able to split this apart when we need to, right? When would you want to do that? Yeah, the replication, cell replication, or when you want to start to make new proteins, right? When you want to actually produce RNA, it's very important to be able to unwind that DNA, be able to pull those apart so that it can actually cause transcription to occur. We'll talk about that in a little bit later. But again, in DNA, the main nitrogenous bases you're going to find here are going to be guanine, cytosine, thymine, and adenine. I think when I think like CGI, CG, that's how I remember those two bind together, and then AT. I don't really know how I remember that. I just remember it's not CG, so it must be AT that bind together, right? So again, and you're going to find this in all forms of DNA. It doesn't really matter where you go to. These are the main 4 nitrogenous bases we're going to be dealing with. Okay. And again, it's very important we don't have a permanent bond there, because we want to have enzymes that can come along and have things like DNA polymerase or RNA polymerase be able to come in and be able to interact with these. And so it's important that bond not really be covalent between these two here. But, right, so anyway, we'll talk more about this in detail in a second. Right, so just some examples of how you can kind of see these forming complementary hydrogen bonds with one another. And again, why you're going to find that when you have mismatches, they don't really go that well together, because again, if you put a C up next to like an A, it doesn't really interact all that well together. These really fit nicely, kind of like, a pair of gloves and a hand essentially. And so, again, based on this sequence here, we're going to get different proteins that can be generated from this, and that's going to dictate function, and which we'll talk about how we can get those proteins formed here in just a minute. Okay, when you're looking at RNA or ribonucleic acid, basically it's just that deoxyribose. You add that oxygen back in, and there you have ribose, right? And so we mentioned the one big difference here is that instead of thymine, we're going to have what? Yeah, uracil is going to be the main thing. It's going to be binding here, right? So again, A is going to bind to U just like A bound to T, but you're just going to find, if you ever see uracil, you're going to know that you're dealing with RNA essentially. Again, just kind of comparing the sugars here. Again, thymine versus uracil. Not important to memorize the structure, but just know that's kind of what the difference is essentially between those two there. Different sugar backbone and a difference in between the thymine and the uracil. Okay, so we'll talk more about DNA a little bit later and replication and all that in the next section. Any questions so far? Fantastic.